Good morning, everyone. I'm delighted to welcome you all today. Today, we'll be exploring the topic of specificity criteria in analytical method validation. My name is Dr. S.K. Singh, and I hold a PhD in this field. In this presentation, we'll cover the key aspects of how to establish and evaluate specificity in analytical methods. We'll look at the criteria involved, common challenges, and best practices to ensure your methods are robust and reliable. So, let's get started and dive into this important subject together. In this slide, we're diving into the concept of method validation, specifically according to USP 1225. When we talk about validation parameters, there are several key aspects we need to consider. First is accuracy, which tells us how close our measurements are to the true value. Then there's precision, indicating the consistency of our results when repeating the test. Specificity is about ensuring our method can accurately measure the analyte in the presence of other components. Linearity refers to the method's ability to produce results that are directly proportional to the concentration of analyte within a given range. Range is the interval between the upper and lower levels of analyte that the method can reliably measure. LOD and LOQ, or limits of detection and quantification, tell us the smallest amount we can detect and quantify with confidence. Finally, robustness assesses how resistant our method is to small variations, ensuring reliability in different conditions. Overall, analytical method validation is about making sure our method is suitable for its intended purpose. It needs to be reliable, so we can trust the results, and scientifically sound, meaning it has a solid basis for producing consistent, accurate data. These parameters together form the foundation for developing and confirming that our analytical methods are fit for purpose in quality control and regulatory settings. Let's talk about the concept of specificity, often referred to as USP or unique selling point. Essentially, specificity is the ability of an analytical method to clearly identify and measure the analyte of interest without confusion. It means we can confidently say, yes, this signal or measurement is coming from the analyte we want to analyze. What's really important here is that this assessment has to be unequivocal, so there's no doubt or ambiguity about the results. Now, to achieve this, our method must be able to distinguish the analyte even when other components are present. These components include impurities, which are unwanted substances that may be present, degradants, which are breakdown products of the analyte, excipients, which are inactive ingredients in formulations, and matrix components, which are the other substances naturally present in the sample. All of these can potentially interfere with the measurement, so the ability to specifically identify the analyte amidst all these possible confounders is what makes a method truly robust and reliable. This is a critical element in method validation because it ensures that our results are both accurate and meaningful, especially in complex samples. So, in summary, specificity is about confidently isolating and measuring our analyte despite the presence of these other components, ensuring the integrity of our analytical results. Let's talk about how we evaluate specificity, which is a crucial part of method validation. Specificity is demonstrated by analyzing several types of samples, including blank samples, placebo, standard solutions, and actual samples. We also test spiked samples that contain impurities, as well as forced degradation samples. This comprehensive approach ensures that our method can distinguish the analyte from other components and potential impurities. Now, why is specificity so important? Well, it ensures that there's no interference at the analyte's retention time, meaning we get a clear and accurate signal for our target compound. This directly impacts the accuracy of our assay results and allows us to correctly quantify impurities. Additionally, specificity is vital for stability indicating capability, so we can reliably detect degradation products. This concept ties into the overall theme of method robustness and reliability. Specificity is especially critical during stability studies, regulatory submissions, and product release processes. Ensuring high specificity means we can confidently assess product quality and safety throughout its life cycle. Now let's look at an example of assay specificity using HPLC. The retention time, or RT, of our active pharmaceutical ingredient, or API, is 5.2 minutes. This is important because it helps us identify the analyte during the analysis. When we talk about specificity for our assay method, we need to ensure that the method can accurately distinguish the analyte from other components. The procedure involves injecting different samples: a blank, a placebo, a standard, and the actual sample. For the blank, we expect no peak at the analyte's RT, which confirms there's no contamination or background interference. The placebo should also show no interference at that RT, ensuring that excipients or other inactive ingredients don't cause any false positives. The standard, which contains pure analyte, should produce a clear peak at 5.2 minutes, confirming our method's ability to detect the analyte accurately. And finally, the sample should also show a clean, well-defined peak at the same RT, demonstrating that our method can reliably detect the analyte in real samples. The acceptance criteria are critical here. We want no co-eluding peaks, meaning nothing else should be coming out of the column at the same time as our analyte. Additionally, the peak purity check must pass, confirming that the peak is indeed from a single component and not a mixture. Altogether, these steps and criteria ensure our assay is specific, giving us confidence that we're accurately measuring the analyte without interference from other substances. This is fundamental for the reliability of our results and overall method validation. Let's talk about the specificity for impurity method. This approach is really focused on how precisely we can identify and quantify impurities in a sample. The key point here is that specificity is all about ensuring that the method can distinguish the impurity from other substances that might be present. This is crucial because, in many cases, impurities can be closely related to the active compound, making their detection a bit tricky. What makes this method important is its ability to give us confidence that what we're measuring truly reflects the impurity of interest, without interference. This ties into the overall theme of accuracy and reliability in analytical methods. So, when we discuss specificity for impurity methods, we're emphasizing the importance of the method's ability to specifically target and measure impurities, which ultimately helps us ensure the safety, quality, and compliance of pharmaceutical products. Now, let's talk about degradation products and their impact on our analysis. It's crucial that these products do not interfere with the analyte peak, because our goal is to accurately measure the active compound without any false signals. This brings us to the importance of specificity. Our method must be stability indicating, meaning it can reliably distinguish the analyte from any degradation products that may form over time or under stress conditions. Speaking of stress conditions, we perform forced degradation studies to challenge our method. These include exposing the sample to various stressors like acid hydrolysis, where we use acid to promote degradation, base hydrolysis, which involves alkaline conditions, and oxidation to simulate oxidative stress. We also consider physical stressors such as thermal stress, which involves heating the sample, photolysis, exposing it to light, and humidity to assess the effects of moisture. By testing under these conditions, we ensure our method is robust enough to accurately identify and quantify the analyte, even when degradation occurs. This is key to demonstrating the stability indicating power of our analytical method and to ensuring the safety and efficacy of the product throughout its shelf life. Now, let's talk about peak purity evaluation using the PDA detector. When we analyze a peak, we look at the peak purity angle, which is a numerical value that indicates how pure the peak is. Alongside that, we have the peak purity threshold, which serves as a benchmark to determine whether the peak is considered pure. The key point here is the acceptance criterion. If the purity angle is less than the purity threshold, it means the peak is pure and not co-eluding with other compounds. This is crucial because it confirms that we're measuring a single, isolated compound, which is essential for accurate quantification and identification. Essentially, a purity angle below the threshold assures us that there are no overlapping peaks or impurities affecting our analysis. This step helps ensure the reliability and validity of our results, tying back into the overall theme of quality control in our analytical process. Let's take a closer look at the difference between specificity and selectivity. These terms often come up in discussions about how agents or systems target their actions. Specificity refers to how precisely a specific molecule or receptor is targeted, meaning it binds to a very particular site. Think of it like a key fitting only one lock. Selectivity, on the other hand, is about preference. An agent might prefer one target over others, but can still act on multiple sites. It's more like a key that fits several locks, but works best on one. Understanding this distinction helps us grasp how drugs or interventions can be designed to maximize effectiveness while minimizing side effects. This concept ties into our overall theme of how targeted actions improve outcomes in pharmacology and therapy. Let's talk about the acceptance criteria for the USB practice, starting with the assay. First, we need to ensure there is no interference at the analyte retention time. This means that when we run our sample, the analyte peak should stand out clearly without any overlapping signals from other substances. Next, the peak purity must pass, confirming that the analyte peak is pure and not contaminated by impurities or degradants. Moving on to impurities, the resolution between peaks should be at least 2. This is a key point because good resolution indicates that the impurities are well separated from each other and from the main analyte, which is critical for accurate identification and quantification. All impurities must be separated effectively, and the degradants should be well resolved from the impurities and the main peak. These criteria are essential to ensure the robustness and reliability of our method, aligning with the overall goal of producing consistent, high-quality analytical results. Let's wrap things up with the key takeaways. Specificity, as we've discussed, is the ability to measure an analyte unequivocally. It's absolutely essential for both assay and impurity methods because it ensures we're accurately identifying and quantifying the substance of interest without interference. We demonstrate this specificity through various studies like blank, placebo, spike, and stress tests, which help confirm that our method is truly selective. Also, resolution and peak purity calculations are critical components in this process. They help us assess whether our peaks are well separated and pure, which is vital for reliable results. And finally, under USB 1225, these parameters are not just recommended, they are required for method validation. So, in essence, ensuring specificity through these approaches is fundamental to developing robust, validated analytical methods. For further support, please don't hesitate to reach out to us. We're here to help and provide any additional information you might need. And with that, I want to thank you all for your attention and engagement throughout this presentation. Remember, we're committed to supporting you beyond today, so feel free to contact us whenever you need assistance. Thank you once again for being part of this session.